Hello everyone. We are excited to welcome you to our synthetic peptide webinar series, where we will lead you through the journey of purification, method development, characterization, and impurity profile monitoring of synthetic peptides. Our webinar today is the first in the series, and today's presentation is entitled Practical Approaches to Peptide Purification. My name is Ashish Chakraborty, and I am with Waters Corporation, and I will be the moderator for today's webinar. With greater efficacy, safety, and tolerability in humans as compared to small molecules, peptides are drawing more attention as potential drug candidates targeting a wide variety of diseases. As investment into these peptide-based therapeutic grows, there is an increasing need for more efficient workflows that can bring this drug to market in a timely manner while preserving the product quality and safety. Let's meet our speaker for today. Joanne Jablonski is a principal scientist in the Separation Technology Division at Waters Corporation. Prior to joining Waters, she worked as a peptide chemist, synthesizing, analyzing, isolating, and characterizing peptides for research, process development, and manufacturing. For the past 20 years, Joanne has provided technical support, training, and application assistance on Waters purification systems. Before our speaker gets started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentation. We will try to answer as many questions as we can, so simply type in your questions in the Q&A box on the left-hand side of your screen, and then press Submit. Upon the conclusion of the webinar, there will be follow-up information sent via email, including a link to the recording of today's presentation. So let's begin our webinar and open the floor to our speaker. Joanne?
Thank you, Ashish. I'd like to welcome you all to today's webinar, and where I'd like to talk to you some of the things that I've learned as a peptide chemist and as a purification scientist working in the field of peptides. Today, we will look at some background information. We'll talk about scaling a separation, how we can optimize a separation if we need to, and then finally, I will end with a practical example where we employ all of the techniques that we've talked about. Everybody knows what preparative chromatography is. We take a crude sample and isolate one or more components from that sample, and that becomes the product that we will take to a subsequent step in a purification process to be used in another experimental study. The purification workflow is pretty straightforward. The first thing we need to do is to determine the amount of material that we need for the next step in our set of experimental studies. To do that, we need to be able to select a column that is of the appropriate size to isolate that amount of material. Once we know what the preparative column is, we can analyze our crude material on an analytical column, and then if we're lucky and our separation is pretty good and we don't need to optimize, then we can do a loading study and scale directly to prep, where we can then isolate our fractions and then analyze them and dry them down. Sometimes we need to optimize the separation because we have closely eluting impurities, or there are other adducts or things that we don't want to contaminate our product. In that case, then we would optimize the separation on the analytical scale and then continue with the loading study, scaling to prep, and isolation. For preparative applications, we want to select a column chemistry that is scalable. There are a number of columns that are suitable for peptide separations. However, the Waters peptide BEH-C18 columns provide the most flexibility for the development of UPLC, UHPLC, HPLC, and lab-scale peptide applications. Waters peptide CSH-C18 columns expand upon the Waters BEH technology in a unique way. The addition of a well-controlled, low-level surface charge makes this particle particularly well-suited for LC-MS applications using non-TFA mobile phases. Waters peptide HSS columns are designed for separations where silica-based selectivities are desired, or if separations are insufficient when using the BEH and CSH columns. This stationary phase provides increased retentivity for peptides and would be a suitable choice for separations using small-scale fraction collection. The estimated peptide mass loading capacity chart is very useful for allowing the peptide scientist to determine which column he needs to select for doing prep. Each diameter has a mass load that is optimum for that particular size column. You'll notice that the estimates of mass load are broad because the capacity depends on the solubility of the peptide in the mobile phase. And sometimes peptides, because they're so complex, are difficult to keep in solution. In addition to the estimated load, each column diameter has an optimum flow rate range and injection volume. So we need to pay particular attention to these values as we're selecting a column to make sure that it will work well with the system that we're planning to use for our analysis and purification. Before I continue, I would like to do some background things that I consider to be housekeeping type things. And that is talking about system volume and impact on prep. System volume is the volume that is from the point of gradient formation to the head of the column. And in a low-pressure gradient system, that would include the proportioning valve and then all of the solvent pathway up to the head of the column. In a high-pressure gradient system, the system volume would begin at the mixer and go to the head of the column. Why is that important? System volume imposes a hold in the preparative gradient to ensure that the same number of column volumes on the prep side as on the analytical side are delivered before the gradient begins. The hold ensures that the peaks from both scales will line up, assuming that all other parameters are properly addressed. So that's a very good thing to have when you have a very complex sample. We want to take away any of the ambiguity that might be present when our peaks don't line up exactly. System volume is pretty easy to determine. The way I do it is I replace a column with a union, and I make my mobile phase A something like methanol or acetonitrile, and then the mobile phase B is that same solvent, methanol or acetonitrile, with a highly absorbing UV compound. You collect baseline at 100% A for about 5 minutes, and then you program an immediate step to 100% B, and then collect data for approximately 5 minutes. Note the time that the curve is at 50% after liftoff. Subtract the programmed method liftoff time from the observed liftoff time. The difference between these two times multiplied by the flow rate will give us the system volume. Take the system volume on the analytical side as well as the preparative side, and I will, once they're determined, post them right on the side of my instrument so that they are readily available for when I do scaling. When we are preparing to do prep scaling, we first need to screen the sample and then do a loading study. A loading study will help us to determine the maximum mass we can introduce to the column and still maintain the resolution between our peak of interest and closely eluting impurities. In this example, the 20 microliter injection still maintains the resolution between our peak of interest. That is the one that is marked by the asterisk and the doublet that follows it. Once we know how much we can load on the analytical column, we can scale to prep. Whether you use the scaling equations and do the math manually, or use the prep calculator available on the Waters website, scaling to prep should give you the same chromatography on the prep side as on the analytical side if you follow all the rules. What are the rules? We use the same chemistry and appropriately scale the gradient, the flow rate, and the injection volume. We don't want any surprises when we go to prep, so if we follow the rules, we should see exactly the same chromatography. So sometimes we need to optimize our separation. And everybody's familiar with the resolution equation. It has both physical and chemical attributes. I won't go through this in great detail, but the thing that's important is to realize that if we double the selectivity, we get the greatest amount of improvement in our separation. The efficiency term can be governed by two things. This is the physical part of the resolution equation, and particle size and column length are the two things that will influence efficiency. Everybody knows that when we go to smaller particle size, we increase efficiency. We also know that if we increase column length, we increase efficiency, but that has a cost, and that cost is a longer runtime. When we have a longer runtime, we also run the risk of having larger fractions, which would impact our dry down time. So these are things that the purification scientist needs to pay attention to as they're planning their separation. Retentivity is one of the chemical attributes of the resolution equation, and things like column chemistry, mobile phase, and gradient are all impacting retentivity. We'll talk a little bit more about the types of gradients in a few minutes, so I won't spend time on those. But we all know that if we change our mobile phases from acetonitrile in water to methanol in water, or change our modifiers, we will change the retentivity of our compound on the column. Selectivity is also impacted by column, mobile phase, and modifier, and sometimes the selectivity can even be more drastically modified if we think about using other types of mobile phases, such as mixtures of acetonitrile and isopropanol, which are especially useful for peptides. We can also change the pH of a solution, go to higher pH using 10 millimolar ammonium bicarbonate or ammonium formate in the middle range of the pH scale, and others. So certainly selectivity is impacted by these three things: the column, the mobile phase, and the modifier. On our website, there are some method development tools that are especially useful. The column selectivity chart helps us to pick different types of column chemistries. So the idea is, if you would like a different selectivity, you pick columns that are far apart on the selectivity chart. Most recently, have introduced the column advisor, which is useful in calling up different columns that meet the attributes that you're wishing to use for your development of your method. We're all familiar with linear gradients, and we can use gradients to optimize a separation. Linear gradients, as we all know, start with low percentage of B and go to high percentage of B in a pretty regimented way. It's in a linear fashion. If we're interested in the peaks that come out at the beginning of a gradient or at the end of a gradient, but we just want to slow it down just a little bit where our peaks of interest come out, we would use a segmented gradient. The type of gradient that I use most frequently for prep is the focused gradient. And there are a couple of reasons for that. I can very quickly get my sample loaded onto the column, stretch out the gradient around my peaks of interest to move those closely eluting impurities away from my peak of interest. And then once my peak of interest is out, I can immediately wash the column and get ready for the next injection. The beauty of a focused gradient is that it doesn't take that much time to get the separation done. So we can very quickly do the purification. So when we focus a gradient, we calculate the volume of the system and the column. And if we've already done our housekeeping things that I talked about earlier, we will have the volume of the system already recorded. We analyze our crude sample with the fast linear gradient, and then using the elution time and the system volume and the gradient table, then we can calculate the percent of organic solvent that elutes the product. Once we know that and we know what the gradient table is, then we can calculate the slope of the generic gradient in percent change per column volume. Typically what we'll do then is to decrease that slope by 5 times, 3 to 5 times usually, and create a shallow gradient segment from about 5% below the calculated elution percentage of our product to 3% above that calculated elution percentage. There's a full description in the application note listed on your screen that goes through each step in great detail. So the result is a focus gradient shown in the bottom trace on this particular slide, where we have decreased our gradient slope from 4.6% change per column volume to 1.1% change per column volume. And you can see for the peak of interest, the one that has the asterisk, this time the third peak in the sample, I have been able to move that particular peak away from the doublet and have almost baseline separation in the focus gradient. You'll notice that I express my gradient slopes in percent change per column volume, and there's a reason for that. It's important to maintain the number of column volumes as we scale from analytical to prep and as we do our focusing to maintain our separation. If you use a gradient slope that is expressed in percent change per unit time, you will make a mistake and your sample will end up in the waste can. So I caution you to use percent change per column volume. Okay, so now let's look at a focused gradient where we apply this technique to a crude peptide. This slide shows a pretty typical crude peptide looking at 220 and 280 nanometers. When I focused the gradient, I noticed that I was able to pull out a couple of small eluting impurities on either side of the peptide of interest at about 6 minutes. Focusing further pulled those impurities even further away, making it more readily possible to get pure product when I scaled up to PrEP. You'll notice that the percent change per column volume is 0.33%. I like to be in the area of about, or in the range of about 0.25 to 0.4% change per column volume when I do peptides. And the reason is because a lot of times peptides are so complex, we need to run a very much slower rate of change. I did a loading study on the analytical column and determined that the maximum amount of material that I could load on it was 10 microliters. The analytical work for this example was done on an Alliance HPLC, and I decided to use the PrEP 150 LC for the PrEP. The PrEP 150 LC is composed of a 2545 pump, either a PDA or UV detector, and a fraction collector. For sample introduction, we can either use a manual injector, as is shown here, or an auto sampler. This system is useful for those who need to purify only a few samples. The PrEP 150 LC is controlled by Chromoscope software, which is very intuitive and simple to use. It has nice features like showing you where the fractions are located in the collector bed and giving you information about the fraction tubes. Because I wasn't really happy with the parameters that I had set for the original collection, I decided to optimize the collection using fraction simulation. You're able to do this by dragging the collection bar on the chromatogram to the areas where you would like to collect. After optimizing the collection parameters, I injected 85 microliters on the preparative column and subsequently 171 microliters on the preparative column with these new fraction collection parameters. And you can see from these traces that the collection is very good. Fraction analysis on the collected fractions was very good, and you can see from these analyses that the collected product was very pure. Sometimes we don't want to scale up, but we want to scale down. The work that you're about to see was done on an Acuity UPLC H-class with a Waters Fraction Manager Analytical, or WFMA for short. The WFMA is a low dispersion fraction collector, specially designed, and this makes collection of small, narrow peaks possible. Fast valve switching and movement between the vessels in the collector enables the collection of these narrow peaks. It has very low carryover and very high recovery. Also, the interior of the WFMA is temperature controlled, which is very useful for those compounds that are temperature sensitive. Scaling from the Alliance down to the smaller column on the H-class, as you can see, gives me exactly the same chromatography. As I mentioned before, when you follow all of the rules, you will get the same chromatography, which is the goal. We don't want any surprises. In this particular example, for collection, I isolated the impurity peak that runs before the peptide of interest, the peptide, and the impurity peak that is eluting after the peptide. Fraction analysis on the collected peptide showed excellent purity at 100%. The purity of the two collected peaks on either side of the peptide of interest also had purities that were quite good. We have recently introduced mass-directed purification with the WFMA. In this system, we simply add a QDA mass detector to the system. The system is controlled by Fraction Links software. Mass-directed purification allows us to more easily identify the peaks of interest and collect only the product of interest instead of having many extra peaks as we would have with UV or other types of optical detection. So we reduce the number of fractions that are collected as well as their subsequent workup. All of this, of course, saves time, and time is of the essence in the purification workflow. Here I have a different peptide where I ran the fast generic gradient in the top trace and then focused the gradient, and you can see that I have very much better resolution between the impurity peaks and the peptide product. I did a loading study and decided to be rather conservative in the amount that I loaded so that I would get pure peptide as a product. Because of the compositional accuracy in the pumping mechanism and the injection precision with the H-class system, I was able to reduce the gradient range even further and run a gradient from 28 to 31% B in 3 minutes. I collected the impurity peaks of interest for further analysis as well as the peptide peak. You can see here that the fractions are collected and are denoted on the chromatogram in 2 different colors, so they're easy to see. And in this particular example, I did several injections and pooled them as I was collecting. As I mentioned earlier, the WFMA has been specially designed to be a low dispersion fraction collector. And here you can see a comparison between the WFMA and a traditional collector and how this special design allows us to collect these very narrow peaks. You can see here that the fraction analysis of those collected peaks was very good, with 98.5% recovery of the peptide product and almost 100% purity. Okay, so sample solubility is very important when we're thinking about doing peptide purifications. In an ideal world, we'd like to dissolve our peptide in the mobile phase, but sometimes that doesn't work because of the properties of the peptide and also because of the other impurities that may be influencing the solubility of the sample. Usually what happens is we either end up with a very large volume of weak solvent for purification, or we end up using a much stronger solvent to dissolve our peptide, and then we end up with distorted chromatography because we all know that strong solvents distort the chromatography in the column. We have our chromatographic pump deliver solvent or mobile phase to the injector where it picks up the sample and is deposited on the head of the column. When we have a strong solvent such as DMSO as our sample solvent on a standard prep separation on a standard gradient system, what happens is the sample is deposited along with the DMSO on the head of the column. The DMSO starts to move down the column and the sample bands move along with it. They don't really interact with the column until they are sufficiently diluted to interact with the bed. When that happens, finally, the sample bands are very wide, and then we end up ultimately with impure product. With a simple plumbing change, we can convert our conventionally plumbed prep system to one that does at-column dilution. With the system configured for at-column dilution, we use a loading pump which is directly connected to the auto-sampler valve to carry the sample through the loop to one side of a T. That loading pump is pumping 100% organic solvent. It bathes the sample with organic solvent on either side of the sample slug. The gradient pump delivers the aqueous mobile phase to the other side of the T. Those two streams meet in the T and very quickly are mixed and deposited on the head of the column. When the sample molecules are deposited on the head of the column in at-column dilution, the DMSO immediately starts moving down the column, leaving the sample as a tight band at the head of the column. Those sample molecules will not begin moving down the column until, in most cases, the DMSO has already been removed from the column bed. When the gradient begins, now the sample bands move down the column and remain as tight bands, which means that now we can get pure fractions. The other advantage to this type of separation is that now we can increase the amount of material that we load on the column. You can see a simple example of a conventional injection of a two-component mix in the top trace, and then I purposely overloaded the column so that you could see what DMSO will do in an overloaded injection on the analytical column. Then I scaled that appropriately to a 19 by 50 millimeter column, and on purpose went ahead and loaded between, I think it was 3 times as much material using at-column dilution. You can see here that the peaks are well resolved and would be very easy to collect. You'll notice that they are eluting a little bit later than the peaks in the top trace, and that is because when we do at-column dilution, we need to load the sample at, we run the loading pump at about 5% of the total flow. So that means that we need to hold long enough to empty our sample loop. So that's why they're eluting a little bit later, but it does not impact the separation in any way. So with at-column dilution, we can sometimes load between 3 and 5 times as much material onto the column. So at-column dilution then permits the large volume injection of strong solvents, and we can improve both our mass capacity and our resolution. We increase our system ruggedness because now our system will not shut down due to precipitation of the sample in the sample loop. And because of the improved system ruggedness, we can also increase our column life. All of these parameters and a lot of the things that I've covered are also available in an application note, which is available for you to peruse at your leisure. We all know that what the effect of temperature is on separations. Sometimes temperature is used to improve peak shape. Some higher temperature is better for a particular sample. Sometimes lower is better for a particular peptide separation. It all depends upon the properties of the particular peptide in question. And while temperature is routinely used in analytical work, it's not so often used for preparative work, and there are a couple of reasons for this. Large diameter columns are not effectively heated from the outside. So while we use column ovens and heating blankets and so forth for analytical columns, they won't work for preparative columns. High flow rate separations really occur at the temperature of the incoming solvent. So if we are using a blanket or an oven on a preparative column, all we're doing is creating a temperature gradient from the inlet to the outlet of the column as well as across the bed of the column. And we all know that temperature gradients impact our chromatography. There is a way to get around this phenomenon, however, and we can very simply add a piece of stainless steel tubing to the inlet of the column and preheat our mobile phase before it enters the column. Then you immerse the column and the sample preheating coil into the bath. This is what it looks like. This is simply a 19 by 100 column with a sample loop at the head of the column. So now we can control the environment, the outside environment of the column, as well as heat the mobile phase before it enters the column. So now you're going to say, okay, I want to do at-column dilution and I want to do temperature control. Not to worry, we can simply put the whole apparatus into the water bath, and we simply put the preheating loop on the side of the mobile phase, aqueous mobile phase that's going into the tea. So now that we've covered some of the techniques that I routinely use to purify peptides, I'd like to share with you a practical example where I employ all of these techniques. The work that you're about to see was performed on an auto purification system that has both an analytical flow path as well as a preparative flow path, and my particular system is fitted with a QDA mass detector. I started my crude peptide isolation with a temperature evaluation at the small scale, and this particular peptide was extremely hydrophilic, and I determined that 40 degrees was the best temperature for this particular separation. I did a loading study using a focus gradient and determined that of 72 micrograms was the maximum load on the analytical column. The area in the red circle shows that as I increase the load, I'm starting to see the presence of a larger and larger impurity peak eluting at the front of the peptide of interest. I did a conventional injection on a 19 by 100 column and collected my peak of interest in fractions 9 and 10. Fraction analysis showed that both of these fractions were very pure, but I was concerned about the small peak that's eluting at around 2 minutes at the beginning of the gradient. Upon further analysis, I was able to determine that it wasn't a system problem or a column problem, but determined that sometimes with TFA in the mobile phase, hydrophobics that are present in the TFA will build up on the column and then elute in the gradient. Did an at-column dilution injection with temperature control. And see here, if we load how much I was able to load on the conventional injection as well as with at-column dilution, I was able to increase the amount of material by about 5 times. I again collected by time, and this time the fractions from 34 to 40 contained my peptide product, able to obtain exactly the same fraction analysis using at-column dilution with 5 times the load as what I got with a conventional injection. In summary then, preparative chromatography is defined by intent, by how much material we need to isolate for those steps in our process which we'll be following. Although purification is traditionally done with UV detection only, mass detection reduces the number of fractions that are collected and decreases post-purification workup. Mass detection also allows us to quickly identify and collect our product peaks, reducing the ambiguity that sometimes accompanies UV-only detection. There are physical and chemical column attributes that improve the resolution. Things like column length and particle size are the physical attributes, and the chemical attributes include modifier changes, pH changes, and mobile phase changes. We can focus the gradient to improve resolution and load and save time. We very quickly are able to bring our gradient to the point where we can focus on only our peak of interest and move those closely eluting impurities away from our product. Temperature control is a useful tool for improving detection, peak shape, and resolution, and can improve the solubility of some peptides. It also allows us to use more viscous solvents like isopropanol or mixtures of isopropanol and acetonitrile, which are especially useful for breaking up peptide aggregates. And finally, at-column dilution permits the injection of large volumes of strong solvents. It improves mass capacity and resolution and will increase system ruggedness by keeping our sample in solution both in the sample loop as well as on the column, and it extends our column life. I invite you to visit the Waters website to peruse the supplementary information available there. Thank you very much for your attention.
That was the warning.
Thank you for your attendance, everyone. We are now ready to entertain some of your questions. So please send those along. We have been receiving some, and we will now answer as many as we can in the time that we have allotted. The first question, do you have any tips for the dissolution of very hydrophobic peptides? I always like to tell people that solubilizing hydrophobic peptides it's really kind of an art, and one thing is not necessarily going to get all of those hydrophobic peptides into solution. Sometimes we can change the order of addition of the solvents that we're trying to dissolve the peptide in. So for example, some peptides you might want to add a little bit of DMSO or acetonitrile first and then add some water. Sometimes it's the reverse. Sometimes we need to break up aggregates. Aggregates of peptides can sometimes be broken up by mixtures of isopropanol and acetonitrile. The thing you have to be careful with, obviously, with viscous solutions is the fact that you may have some overpressuring. So that would be an instance where temperature control would be useful. When you have extremely hydrophobic peptides and you cannot get them into solution no matter what you do, I typically use solvents like DMSO. The DMSO will elute early in the gradient, and hopefully your peptide with temperature control, if you need it, will stay in solution so that you can get the separation to complete. Okay. What is your experience with Hillock for the purification of polar peptides? Hillock is only recently come into common usage. We do have an application note that is available, and I would recommend that you take a look at that. It's on our website. I can give you the reference number. It is 720004293EN. Again, 720004293EN. question. Do you advocate using temperature control for all separations? Not necessarily, only those separations where it might be needed. Sometimes it's necessary to keep a peptide in solution, especially very hydrophobic ones, and sometimes that ultimately aids in the detection of the peptide. Although the method that I discussed works quite well, it is rather unattractive, so I always advocate trying to do separations at room temperature first and use heating only if it's absolutely necessary. Next question. Please comment further about the effects of particle size on preparative chromatography. Okay, so typically when we go to smaller particle size, we will get better resolution. And also, we all know that when you go to smaller particle size, we also run the risk of increasing our overall system pressure. So it's a balancing act with particle size. Typically for the range of peptide separations and purifications that we do, we're at the lab scale, which is a few milligrams to hundreds of milligrams. And I typically use a 5-micron column or a 10-micron column. Of course, you want to use the appropriately sized column that will be the best one for the amount of peptide that you need to purify. So the shortest column is best because it will take less time to purify, and also your fractions will tend to be a little bit smaller. Okay, next question. I missed what you said about the last slide about breaking aggregate. Okay, so sometimes when we put a sample or try to get a sample into solution, it clumps up and it's hard to break up those aggregates. I've found that using mixtures of acetonitrile and isopropanol helps to break up those aggregates. Next question. How do you differentiate a peptide with the same sequence but different chirality? I would just begin using a chiral column to see if you can effectively separate those two peptides. Can you use DMF as well as DMSO? Absolutely. That's not a problem. A lot of times you just have to see what will work. Remember, every peptide has different properties because of the sequence and also because of how it's worked up. During the synthesis and the cleavage, we can end up with a whole host of adducts and contaminants that also influence how well we can get our peptide into solution. So all of those, in conjunction with the character of the peptide itself, are going to impact its solubility. So yes, you can use DMSO as well as DMF. The next question is, could you comment on pore size and separations? When would you use 300 angstrom versus 130 angstrom? Generally, we use the 300 angstrom for very long peptides. We did do a study a couple of years back where we compared 130 and 300, and the difference was minimal. However, a safe bet is to go with the 300 angstrom for very long peptides. I would say, in 30 or 40 mers and up. Define very long peptides, please. Okay, so I would say long peptides would be those that are over over 25 or 30 typical. I think of peptides that are of average length are somewhere between 15 and 20 amino acids in length. Is there any major difference between purifying synthetic versus natural peptides? As far as I'm aware, not really. I think that obviously you need to know what the properties of each peptide is before you begin, and then take a note as to whether they are acidic or basic or hydrophobic, and proceed as you would, as you normally would. Is it possible to separate a racemic mixture of peptides with a reverse phase column? The answer to that is not really. Sometimes you will need to employ other methods to affect the separation. Can we use a 130 angstrom for a long peptide? Yes, you can. That shouldn't be a problem. You will be able to tell when you do your analytical work. Usually before we go to a preparative column, we will do some analytical screening, as I mentioned before, and you will be able to tell if that 130 angstrom particle pore size will be adequate for separation. Next question. At column dilution, if you're unable to replumb the system, can you inject a blank of DMSO followed by the sample, followed by a blank of DMSO? That's typically what we call sample sandwiching, and it's possible to do that. In effect, what you're trying to do is to keep the sample in solution in the sample loop so that it doesn't precipitate. And one of the things that column dilution does, obviously, is to prevent sample precipitation in the injection loop. And it does that by delivering 100% organic solvent, because that's plumbed directly to the injection loop. And the sample, therefore, has organic solvent sitting on either side of it as it's sitting in the sample loop. So this is more of a sample sandwiching is more of a manual type process for trying to keep your sample in solution. However, when you do the injection, you do run the risk of having that sample precipitate on the top of the column, because now it's not being diluted slowly over the course of time like it would be with that column dilution. Okay, next question. You mentioned that temperature control helps in improving resolution. May I know either whether increasing temperature will reduce the lifespan of the column? Okay, so the first thing that you need to pay attention to, obviously, would be the parameters that your column manufacturer puts forth for the particular chemistry, column chemistry that you're using. Typically, columns will be pretty safe in the middle range. I would say anywhere from 30 to 60 degrees. Some may be able to go higher, but definitely I would check the manufacturer's recommendations before using too high of a temperature. Okay. The last question is, you mentioned doing a loading study. Can you explain how to do a loading study? And the answer to that, of course, is yes. Loading studies can be performed at either the analytical or the preparative scales. However, I prefer to do them at the analytical scale to save on the sample. And you can either perform this by increasing the injection volume of a concentrated sample solution and then running the proposed HPLC method that will be used for the separation, or several samples of the same compound in varying amounts can be dissolved in separate vials using a known volume of the diluent. So these individual samples then can be injected and run using your HPLC method that will be used for the prep, but on the analytical column. The idea, obviously, is to maximize the amount of sample that can be introduced to the column without the loss of resolution between your peak of interest and its closely eluting contaminants. And when your resolution is lost, we know that we've loaded too much sample and can lower the injection amount. And that's the amount that we scale for the preparative run. Okay, so now we're out of time, and this brings us to the end of this webinar. Thank you all very much for joining us. The next webinar in the series will be on developing quicker and better analytical methods to separate API from impurities, and will be presented on Monday, July 9th. So please register at www.waters.com/synthetic peptide webinars if you haven't done so already. We look forward to seeing you again soon. Thank you very much for your attention.